Okay, so welcome back. Welcome back, you guys. And so today, I think is perhaps one of the more interesting topics we're going to be covering, next generation sequencing and variant discovery. But before we dig in, are there any questions about other material we've covered? Okay. All right. We got a lot of material to cover, so I think I'm going to just dive right in today. We're winding down. Next week is genetic engineering. Then we're going to have a clinical genetics informatics class. By the way, if there's this clinical genetics informatics class is a new module. If there's specific topics in informatics that you were hoping to see covered in that class, if you could let me know. I'm still kind of finalizing the agenda, the table of contents for that particular module. And so please do let me know if there's any specific topics you'd like to see covered during that session. And then we'll have our ethics debate. Okay. For today, we're going to delve right into the clinical case. What we're going to do is present a case of a guy, 38-year-old guy that had a heart attack, and he's curious to know, is his heart disease hereditary, and if so, what mutation would have caused it? And what we're going to wind up doing is walking through the whole process end to end of discovering, trying to figure out exactly what mutation in this patient caused his heart disease. And we're going to examine it really from two perspectives. One is the next generation sequencing. What are the technical and bioinformatic aspects of discovering variants and filtering it down to identify the key causative variant? And then we're going to also examine it subsequently from a clinical perspective, which is having believed you've focused in on the causative variant, what does it take to actually deem a particular variant to be pathogenic? Today's homework assignment is going to be, this is a hard homework assignment where I'm going to give you 4 variants and ask you to go off and try to discover information about these variants, their clinical significance, the disease they cause. And so I'm going to describe the homework assignment at the end, but maybe be thinking about the homework assignment as we go through it. I'm going to suggest that you guys pair up to work on this homework assignment, because I think that this is going to be a pretty challenging homework assignment. But I think that if you do it, if you spend the time to do this particular homework assignment, it's going to wind up kind of forcing you to be exposed to a lot of the informatics and bioinformatics resources that we will have been talking about. And my hope is that the hour you invest in this particular homework assignment, you'll feel that it was well worth it. So kind of be thinking about that as we're going through this. I'm going to be asking you to take a set of variants and determine their clinical significance, and be thinking about, who in the class you might want to pair up with, because I think the assignment is going to be a little bit challenging, and if you work in a team, then I think you may wind up benefiting a little bit more. Okay, so let's start. Our case today is a 38-year-old male, and he comes into the office, and he was previously well, but he recently was hospitalized with an acute myocardial infarction. And during that hospitalization, he was found to have a markedly elevated cholesterol. He is sedentary, and he smokes a pack of cigarettes per day. So in the office, he's examined. He's mildly obese with a body mass index of 30. And we notice this on the back of his Achilles tendon. And what this is a xanthoma. And it's a lipid deposit often seen in cases of hypercholesterolemia. And this guy was found to have elevated cholesterol when he was in the hospital. And so we measure his cholesterol level, and we find that his LDL cholesterol, or the so-called bad cholesterol, which ideally is going to be around 100, in this guy is markedly elevated. We find that his LDL cholesterol is 350. So he's a young guy that had a heart attack, has a very high cholesterol. He's sedentary, he's a smoker, he's a little bit obese, and he's got evidence of markedly increased LDL cholesterol. And when we look at his family history, we see that his brother also has coronary artery disease and has high cholesterol. We find that his mother has coronary artery disease. We find that his maternal grandmother and his two maternal uncles both died of coronary artery disease. So it does look as though he has a hereditary type of heart disease, and that, coupled with his markedly elevated cholesterol, leads us to suspect that he may have a condition known as familial hypercholesterolemia. Okay, familial hypercholesterolemia. The main questions that this patient has as he's sitting there in the office is, was my heart attack genetic? What is the risk of passing this to my offspring? And what mutation do I have? So the first question, is my heart attack genetic? Well, based on family history, without knowing anything more, I think we could say yes. There's without a doubt a genetic component. And in fact, when you look at the family history, it appears to be inherited in an autosomal dominant inheritance pattern, in which case the risk of passing this to his offspring would be the risk you'd see with any autosomal dominant inheritance pattern, which would be 50% of his offspring would inherit whatever it was in this case that caused him to have this premature heart disease. The next question, though, what mutation do I have? We suspect he has familial hypercholesterolemia, but now let's go ahead and see if we can figure out exactly what mutation this patient has. To do that, it helps to know a little bit more about what we suspect is going on, familial hypercholesterolemia, because doing a little bit of an informed search of the genome for a variant will be far more fruitful than doing, if we simply look at this guy's genome and look at all the variants and we find that there's 4 million variants in his genome, it's going to be a little bit less guided than if we are at least kind of sort of looking for something. So it helps before we go exploring this patient's genome to be kind of informed about familial hypercholesterolemia and kind of be a little bit directed in our search. So familial hypercholesterolemia, we know can be caused by 4 different genes. By far, the most commonly mutated gene is the LDL receptor gene. What's happening here, and imagine that this is a liver cell that you see in this picture, and this picture is illustrating the way in which the LDL cholesterol, the bad cholesterol, gets taken up by the liver cells and processed. You have these receptors on the surface of the liver cells. These receptors, or these LDL receptors, bind to the LDL cholesterol that's circulating in the plasma, and they take that LDL cholesterol up into the cell. These LDL receptors are proteins. And so, like other proteins, they're initially produced. The ribosomes are reading the RNA and producing the LDL receptors here in the endoplasmic reticulum. The protein in the endoplasmic reticulum buds off and fuses with the Golgi complex, and within the Golgi complex, the LDL receptor protein is further modified, and the modifications then direct that protein through these vesicles to fuse with the cell membrane and expose the LDL receptor. So this LDL receptor protein is the product of the LDL receptor gene. The LDL cholesterol is a cholesterol ester core surrounded by a specific lipoprotein called B100. So this is an LDL particle that would be circulating in the plasma. And this LDL particle binds to the LDL receptor. The protein surrounding this particular cholesterol ester core, this apolipoprotein B100, can also be mutated. And if it is mutated, it may no longer bind to the LDL receptor. And so we also see that mutations in apolipoprotein B100 gene can increase the LDL cholesterol level because it interferes with the ability of the LDL particle to bind to the receptor. When the LDL cholesterol binds to the receptor, these LDL receptors that are bound to the LDL coalesce in these pits. These pits are lined with a protein called clathrin. So these LDL receptors bound to the LDL particles collect in these clathrin-coated pits. There's another protein produced by the ARH adapter protein gene that promotes the clustering of these LDL receptors in these clathrin-coated pits. Finally, once you have the LDL receptors and the LDL in these pits, this becomes a little vesicle that's taken up into the cell, and that allows the cell to then absorb the cholesterol. But then the LDL receptors get cycled back to the cell surface. And you can have a mutation in the PCSK9 protease gene that interferes with recycling of the LDL receptor. So these four genes have been associated with familial hypercholesterolemia, and they all work by interfering with the process by which the LDL receptor binds to the LDL cholesterol and takes it up into the cell, up into a hepatocyte, for instance. And we can see this again on this picture. We're now looking at the LDL receptor in a little bit more detail because, again, I think it helps to understand the pathophysiology of familial hypercholesterolemia, which is what we suspect in this patient, as we go off trying to search for his particular mutation. So we see here that the LDL receptor is being produced. It buds off from the endoplasmic reticulum, it fuses with the Golgi complex, and it makes its way to the cell surface. There are some mutations in the LDL receptor gene that are so bad that they lead to complete absence of any LDL receptor protein product, so-called null alleles, or these class 1 mutations. These would be the most severe because you would wind up getting essentially no receptor synthesis. We also kind of looked at different classifications of mutations. You may recall when we talked about cystic fibrosis. Class 2 mutations of the LDL receptor interfere with moving the protein product from the endoplasmic reticulum to the Golgi complex. We then have class 3 mutations that interfere with LDL binding by the receptor, and etc. We have a number of these different classes. There's over 1,100 known mutations, and again, the worst one here would be the class 1 mutations, which would result in essentially no protein product, which would result in no LDL receptors. Just for interest, a little bit more of this picture before we go on. When the cell does take in, so here you have these LDL receptors that are bound to the LDL particles, and now they're all collected in these clathrin-coated pits. These coated vesicles are taken into the cell. The LDL receptors are recycled. They go back to the surface of the cell. The LDL cholesterol then is split apart. The protein, the apoprotein B surrounding the cholesterol moiety, is broken down into its constituent amino acids, and then the cholesterol, the free cholesterol, is released into the cell. And here you can see, out of interest, it decreases further production of cholesterol within the cell by inhibiting HMG-CoA reductase. So it further also decreases LDL receptor synthesis, decreases cholesterol synthesis, and it also increases this acyl-CoA cholesterol acyltransferase enzyme, which allows the cholesterol esters to be collected in droplets in the cell. And then these, of course, decrease new cholesterol formation. So LDL receptor, the LDL receptor, the gene for the LDL receptor has over 1,100 known mutations. About 10% of them are large insertions or deletions. And a couple of things that are important to know about this particular gene, and to kind of emphasize why, the more you know about the disease you're pursuing, the more likely it is that you're going to be able to discover the variants. For this particular gene, and this is the most common cause of familial hypercholesterolemia, even though those other 3 genes can be associated with the disease. Mutations in the LDL receptor gene are the most common. A lot of these mutations are what are known as private mutations. And a private mutation is where you see a mutation in a particular family that hasn't been seen elsewhere. And this is, like, if you look at cystic fibrosis, you may remember that there was one mutation in particular that was very, very common. And that's in counter-distinction to a private mutation, which is very rare. It's seen within a particular family, and that same mutation is generally not seen in other families. So a lot of different private mutations, and as you might imagine, that might make searching for a particular mutation tricky, because if this patient has a private mutation, it might be that it's a mutation we've never seen before. It's not that we're not going to find in any public registry. The other thing that makes this gene interesting is that not all of the mutations are functionally significant. there's a lot of mutations in this gene that occur in areas where, in an intron, for instance, that have no functional significance, or there's a mutation that's a synonymous mutation that doesn't change the protein product. And so we can't simply go on a hunt for all of the different variations in this individual patient's LDLR gene, because we may come across a number of mutations, and we still don't necessarily know which one is the significant one. The other thing that's important to know about the LDLR gene is that about 10% of the mutations are large insertions or deletions. And we talked about this a little bit last week. We'll be discussing it more this week, but the next generation sequencing may miss large insertions or deletions. So here it is. We suspect that this patient has familial hypercholesterolemia. We suspect, therefore, that he's got some type of mutation in one of these 4 genes. Statistically, most likely, he's going to have a mutation in the LDL receptor gene, but we know that that gene has private mutations. We know that 10% of patients have variations that may be missed on next-generation sequencing, and we know that not all mutations are functionally significant. So given that, we have to decide how we want to approach testing this person to begin exploring his genome to look for variants. And so one of the questions that comes up is, well, what test should we do? Or what test might we do next to look for known pathogenic variants in any of the 4 genes and large insertions and deletions? And you may remember from last week that the DNA chip technology offers a strategy here. Number one, it's very inexpensive. You can design a chip that has probes for all of the known variants, all of the known pathologic variants in these particular 4 genes. And you may also recall from last week that the comparative genome hybridization chips are very good at detecting duplications or deletions, or what are called copy number variants. So we began in this patient by looking for known pathogenic variants using a SNP array, and we also look for large insertions and deletions using a comparative genome hybridization array. So again, here we have a chip where we have probes for all of the known pathogenic variants of these particular 4 genes, and we examine for copy number variants using comparative genome hybridization. And in this particular patient, of course, just because we want the case to be fun and illustrative, he winds up not having any notable findings on his chip assay. Okay, so none of the known pathogenic SNPs appear to be found, and there's no evidence that he has any large deletions or large duplications. And yet, the guy's 38 years old with a very compelling family history for an autosomal dominant type of coronary artery disease, and his LDL cholesterol is sky high. So we're still strongly convinced that he has familial hypercholesterolemia, and we know that this particular LDLR gene has a lot of private mutations. So we begin to suspect that he may have a mutation, a point mutation, for instance, in one of his genes that we've never seen before. And in order to explore that, we then embark on whole genome sequencing. And so really, what this class is devoted to is whole genome sequencing. And we're going to walk through the process now of applying whole genome sequencing to this particular patient. Really what we're trying to do is figure out in this particular case what mutation is responsible for this patient's familial hypercholesterolemia. And I broke this whole genome sequencing down really into 2 steps. One is the technical / bioinformatics process of looking at this particular patient's DNA and just cataloging the variants that we see. And then next is also a bit of an informatics, bioinformatics clinical process where we take that list of variants and we try to figure out which ones are clinically significant and which one in particular may be the culprit pathogenic variant in this case. What we're going to do for the next handful of slides is go through the bioinformatics process whereby whole genome sequencing yields up a variant list, okay? So let's see, here we go. Okay. And my guess is that for a lot of you, some of these file formats and some of the standards that are part of the bioinformatics community will be new, but I also think it's just kind of super interesting to understand the whole pipeline process of next generation sequencing, in part because it'll help you get a qualitative sense of the quality and reliability issues. And I think that, people can naively think, oh, you did next generation sequencing, great. That means I have your 3 billion base pairs all neatly in a row. And I think walking through the technical aspects and the bioinformatics aspects, I hope will give you at least a qualitative sense of, oh, there's some quality issues there. It might be that this pipeline leads to a list of variants, but how reliable is that particular list of variants? So what we're going to do is show, okay, we're going to perform next generation sequencing on the subject specimen. That's going to yield a file that has all of those reads, and that particular file is going to be in FASTQ format. We're going to compare those reads against the reference sequence from the NIH, and the reference sequence is in a file format called the FASTA format. And for each one of these reads in the next generation sequencing, we're going to align it to the reference. And the alignment between each one of these reads and the reference sequence, all of those alignments will be put into a file called the sequence alignment map, and that's in SAM format. And once we have all of those reads aligned to the reference sequence, we can then generate a list of variants, and the list of variants will be in a file called a VCF file. And you may remember last week we introduced this notion of next generation sequencing, where you take the DNA and you chop it up into a lot of little pieces. And each one of those pieces is sequenced in parallel. And you wind up with a lot of short reads of DNA sequence. Depending on the technology, each one of these reads might be 100 base pairs long, maybe several hundred base pairs long, depending on the technology being used. And a file that contains the sequence of each one of these reads can be generated, and that file format would be a FASTQ file format. We then align each one of these reads to the reference genome, and the reference genome can be obtained in FASTA format. And when we align each one of these reads to the reference sequence, as you'll see is done here, this file containing all of the reads aligned to the reference sequence is in SAM format, or sequence alignment map format, okay? So first thing we do is next generation sequencing on the subject specimen, and we wind up getting a file that's in FASTQ format. And you'll see here a snippet of a FASTQ file. And in the slide deck, there's a reference for where you can learn more about the FASTQ format if you'd like. And one of the things you'll see here is that the FASTQ format there's 4 lines for each read. Okay, so one of the reads is 4 lines. Then here's another read, and it's got 4 lines. And then there's another read, which is 4 lines. The first line is an identifier about this particular read. The second line is the actual sequence of base pairs in the read. The third line is just comments. And then the fourth line is each one of these ASCII characters corresponds to a quality score that corresponds to the particular base. And one of the things you'll notice here is, in some cases, the particular sequencer couldn't tell what base was at this location and simply inserted an N for nucleotide. The quality scores, the per base quality scores, each ASCII character, again, corresponds to a quality score. Generally, a quality score of 10 to the minus third error rate is set at a minimum for high quality reads. What that means is an error rate of 1 in 1,000. So if we set as a minimum an error rate of 1 in 1,000, that means that when we call a base of A, for instance, we're 99.9% sure that this is the right read. Okay. Trying to give a quantitative description of the overall quality of FASTQ, it's a little bit tricky because in the end what's going to happen is a lot of these reads are going to be stacked together in the sequence alignment map, and the overall quality, the overall certainty by which you call a base is going to be dependent on a number of different factors. So what these numbers here, what these quality scores here represent is the per base quality score for a particular read. And again, each one of these ASCII characters corresponds to a different quality score. And usually the bar is set at least 1 in 100 or 1 in 1,000 or 99% accuracy at this stage, or depending on the scenario, a 99.9% accuracy is required at the read stage in order to have any confidence in this base, in order to use it for further processing as we go downstream. So now we've taken all of those reads in the FASTQ format. Possibly we've filtered out the ones that have a poor quality score. And now each one of these reads, so each row here represents an individual read. And we now are aligning each one of these reads to the reference. So this would be the reference file. And this file that you have here now would be a sequence alignment map. And this particular file can be downloaded in a SAM format. Okay. And the number of reads for a particular base can vary quite a bit depending on the technology. This is an experiment where this was whole exon sequencing rather than whole genome sequencing. And so there were fragments, DNA read fragments, just corresponding to the exons of a particular gene. And so what you'll see here is that there's a tremendous number of reads overlapping the exons and almost no reads overlapping the introns. And that was the way that this particular experiment was designed. And in this case, this peak here, this is just showing how many reads there are at a particular spot. And this particular peak here, there was 1,000 reads. So at this particular base, for instance, 1,000 different fragments overlap that particular base. And as you might guess, and I'm not going to get into it quantitatively, but at least qualitatively, as you might guess, where you have a lot of reads, you probably can call the base at a particular spot with greater confidence, right? Because if you have 1,000 reads, and even if one or two of those bases differs, if 999 of those bases say one thing and one base says something else, you're probably pretty sure what that base is. And in fact, going back to this slide here, if you notice right here, we have one of the reads called the base at this position a T, but all of the other reads called this a C. And so even if the accuracy of the reads was only 1 in 100, because there are so many reads here, we're pretty confident that in the patient's DNA sequence at this particular locus, this patient has a C, and that the T was some type of artifact. Okay. So you'll see here that depending on and sometimes these whole exon sequencing experiments will have a lot more reads per locus than whole genome sequencing. For whole genome sequencing, you may have, for instance, 50 to 100 reads per spot. But the more reads that you have at a particular spot, the more confidence you have in the base that you're calling at that particular spot. In this particular case, a variant was found at this particular base right here, but you'll also notice that this variant was called at a spot where there was very few reads. And so in this particular experiment, even though a variant was identified here, it was rejected just because there wasn't very many reads at that particular spot. So the SAM file, and there's a lot of tools for viewing SAM files and drawing inferences from SAM files. And what we're going to be talking about next is how the SAM file is an input into the software that then begins to compute the variance. Okay. I think it's actually kind of impressive when you think about, the amount of data that's generated when you do a next generation sequencing run, and how the next generation sequencing run on one person can generate a terabyte of data. And when you begin to see, oh, okay, I get it now. It's because you're not just getting one long chain of bases. Really, when you do next generation sequencing on someone, what you're getting is just millions and millions of fragments, millions and millions of reads. And each one of these reads has to be aligned. And so you have just a tremendous amount of data here. You'll see that there's some pink strands and some blue strands, and those represent forward and backward reads. Sometimes when you have a fragment of DNA, you can read it going from left to right, but you can also read it going from right to left. And these paired end reads can be very useful in helping to identify different types of variants, which we'll talk about in a minute. But paired end reads are commonly employed in next generation sequencing, where you take a particular fragment of DNA and you're reading it in both directions. So let's say we've generated our sequence alignment map, and we now have all of these sequences lined up against the reference, and here's the reference sequence down here at the bottom. And in this particular case, you'll see that for all of the different sequences that aligned, they all have a C at this position, whereas the reference has a T. And I think you can understand how it would be possible for a software algorithm to infer from this, okay, the individual whose DNA we're analyzing obviously has a SNP, has a polymorphism or has a variant at this particular locus. You know, they have a C instead of a T. And you might also infer from this that it's a homozygous polymorphism, you know, that both of their chromosomes have a C, whereas the reference DNA has a T. So looking for the SNPs, that seems to be pretty intuitive, how you could use software there. Sometimes one of these might not be a C because it might be an error, and the software has to accommodate, you know, at what point do you say it's an error, at what point do you call it as a heterozygote versus homozygote, etc. Likewise, you can see here all of these SNPs kind of sort of line up to the particular reference DNA, although they all appear to have a one base pair deletion with respect to the reference DNA. And so it looks as though this particular patient, which happens to be a dog, because it was the best example I could find, but this particular patient appears to have a one base pair deletion because that's what we see when we line up all of the fragments. And so you could imagine how a computer algorithm could look at all of these alignments and say, you know what, this particular fragment lines up to the reference, although the reference had a G right in here, which we don't see. So I can infer from that that this fragment must have a one base pair deletion. And we see that consistently when we're aligning all of these. And so you can begin to imagine how you can take this sequence alignment map and apply software and generate a list of variants. And for the SNPs and for the small insertions and deletions, it might be somewhat intuitive how to infer those particular variants. But when we start talking about larger deletions and inversions and mobile element insertions and tandem duplications, the algorithms that are used to infer those become far more complex. And I kind of sort of only brought this picture in, not to explain it, but only to kind of have your gut reaction of looking at this and saying, well, that looks hard, right? And what this picture is trying to show you graphically is how various types of computer algorithms look at sequence alignment maps and infer different types of variants. And for instance, read depth, if there's not very many fragments, or if there's no fragments overlapping a particular region of the reference sequence, you might infer that there must have been a deletion in the patient sample. Ooh, who's making that scratchy noise? On the other hand, if we see a lot of extra reads piling up corresponding to an area of the reference, we might infer from there that there's a duplication. These split end reads or the read pairs can also be effective in pointing out that there's deletions. If you start reading this particular fragment from left to right, and it aligns with one portion of the reference DNA, but when you read it in the other direction, it aligns with a different portion of the DNA that's a lot farther away than it is in the fragment, you can infer that there was a deletion. So again, not to go into this slide in too much detail, other than to kind of give you a warm fuzzy or a qualitative understanding that it's not a straightforward process to look at a sequence alignment map and simply say, here's all the variants. There's complex software that's involved in the process. And as you might guess, because there's complex software involved in the process, and we'll come back to this in a little bit, there can be reliability issues in the final list of variants that are called, more so with the more complex variants, but even with the smaller insertion deletions and SNPs. Finally, we wind up with the VCF file, and this is just a long list. In our patient, this is a file that has 4 million rows in it, 1 row for each variant. Every place where the patient's DNA differed from the reference DNA, you wind up getting another row in the VCF file. And the VCF file, it has a header that has some descriptive information, and then there's 1 row for each variant. And so here is an example. On chromosome 1, at this particular position, the reference DNA had a G, but in our particular patient, we found an A. Okay, so this would be a simple SNP. And so the types of things that you'll see in these VCF files will be SNPs. So for instance, at position 2, the reference had a C, but we found a T. Or you might see an insertion. So position 2, the reference had a C, but we found a CT. So there was a T inserted. Or for a deletion, at position 1, there was an ACG, but we just saw an A. So the CG are missing. Or for a replacement, at position 1, there was an ACG, but we just saw an AT. So the CG are gone, but a T is in its place. Or the VCF file might contain information, a row about a large structural variant, where, for instance, at position 100, the reference had a T, but what we see is that everything is gone up until position 299. Okay, so a very large deletion. And again, the output of these next generation sequencing pipelines list all these variants. You wind up with this huge list of variants, and then it's from there a matter of looking at these variants, trying to figure out which one is potentially clinically significant. Yeah.
Does the reference sequence represent the accumulation of knowledge about what is normal or most common? You might imagine any original source reference sequence has its own mutations, variants, and SNPs. How does that work?
It's probably more correct to say most common as opposed to normal. But what happens is, so the NCBI, for instance, maintains reference sequences. And what they do, for the most part, is they look at as much examples as they can. And in the reference sequence, it really doesn't correspond to any one single person, but at any particular locus, the most common base would be present. And I think the last reference sequence that was published by NCBI is now 3 or 4 years old. And they have several reference sequences prior to that. And in some sense, what's more important is knowing what the reference sequence is, because these the VCF files will always reference, here for instance, I'm looking at this chromosome and here's the reference sequence that I'm using right here, NM173808, and you can go to the NCBI site, and you can plug in that identifier, and it'll show you the FASTA file for that particular reference format. And so for the most part, what these reference sequences represent are the most common base at every position along the way. I think every 3 to 5 years or so, NCBI plans on updating their reference sequence to be the most accurate that it can. And here I think, from a clinical perspective, it helps to get a warm, fuzzy, qualitative understanding of the reliability of variant calls. And it's difficult to give a quantitative assessment because it's very machine dependent, and the actual accuracy of a specific variant call depends on a lot of different factors, not just the reliability of a particular base call, but it also depends on the read depth. It depends on the correction algorithms. And so it's very difficult to give an overall accuracy. For the most part, what a lot of companies claim, their overall accuracy for a call at an exon of interest, might exceed 99.999%, whereas across the board it might be smaller. But you have a number of challenges when you're trying to determine your particular list of variants. When you're performing next generation sequencing, you have repetitive sequences that are challenging, duplications and deletions. Homopolymers are tracks of identical nucleotides all in a row, and depending on the next generation sequencing technology, it can be confusing how many particular bases, like bases all in a row. Long read lengths can pose a problem, areas where you have increased GC content. And I think it's interesting to note that 5% of the human genome has yet to be sequenced. Despite the fact that people have published, that we know the complete sequence of the human genome, 5% of it remains to be sequenced. When you then take the subject's FASTQ file and then go and try to align it to the reference sequence, additional challenges arise. Repetitive regions, you may recall that roughly 50% of our DNA is repetitive regions. And so imagine if you have a small read of 100 bases and you now want to figure out where does that particular read map to the reference. Well, if it happens to be a read from a piece of DNA that's replicated multiple times across the genome, it's going to be very difficult to know exactly where it goes. On the other hand, the repetitive regions tend to occur in the regions between the genes. So we don't see as many repetitive regions. We don't see anywhere nearly as many repetitive regions in the genes. So again, you see all of these challenges, but the quality of the readings from the exons tends to be very high, but across the board, there are still some reliability issues. And when you're then looking at the sequence alignment map and trying to develop the variants, the list of variants, just from this slide here, you can just imagine, geez, this can't be something that's 100% accurate. And in fact, different software methods wind up generating different lists of variants. Read depth can be taken into account. There's lack of concordance across algorithms. Some variant callers don't look at contiguous variants. So if you have a gene that has one variant in it, that'll be called, or if you have another piece of software that might look at, well, here's all of the variants within a particular gene, that corresponds to, for instance, a different allele. This textbook, the bioinformatics textbook, I just pulled this quote out only because it was the only time he used the word extraordinary in the whole book. And, so many things are extraordinary when it comes to genetics. But he says that calling variants and interpreting their significance are extraordinarily complex. And then another interesting article in 2013, this guy compared 5 variant calling workflows and found only a 57% concordance with 5% of the variants unique to each pipeline. So again, I'm going to kind of leave it at this. I wanted to just kind of leave you with a qualitative sense, at least, of the complexity involved in, it's not just, oh, let's do next generation sequencing, and boom, you have this nice pretty list of 3 billion bases, and that's your genome. Rather, what you have are these sequence alignment files, and you have these VCF files, and they have some inherent challenges in them, and so they have to be taken with a little bit of a grain of salt. And that's why, for instance, you'll find in clinical use, when you look at some of the testing that's done with next-generation sequencing, you'll often find that the results of next-generation sequencing are actually being confirmed using a process of Sanger sequencing, which is going to be far more precise. Given the challenges with reliability of variant calls, a lot of labs will have adopted the genome analysis toolkit best practices pipeline. And what that does is it defines a process of going from the reads coming out of the sequencer all the way to calling variants. It describes the QA steps, it describes the software to be used, and the objective of the genome analysis toolkit is to try to standardize the variant calling process to improve the consistency that you see. If you give these specimens to 2 different labs, to try to approach the point where you're getting back the same list of variants from different sources. Okay, so we've now generated a variant list such as it is. And in our particular patient, we have about 4 million variants. And, for the most part, we have some confidence in those variants. Again, it might be that ultimately we do some confirmatory testing on some of those using different techniques. But we've generated the variant list. There's 4 million of them. That's a mouthful of variants. And it would make a very big report if we were to send that back to the clinician. So what we're going to do is assess those variants now for clinical significance. And we do that using different types of filtering approaches. And I only show you this one. I'm not going to go into any detail on this particular filtering approach, but I show you this one because this was a filtering approach that was used in a clinical practice for patient care. It's out of a New England Journal of Medicine article in 2013. They used a whole exon sequencing approach to diagnose a range of conditions in 250 consecutive patients at Baylor. And they did establish a diagnosis in about 25% of cases, but also they confirmed all findings with Sanger sequencing. So they started off by having 200,000 to 400,000 variants. We have 4 million because we're doing whole genome sequencing, and this was based on whole exome sequencing. And they went through a particular filtering to ultimately try to identify the causative variants. And I think that, again, I show you this because it was used in real clinical practice. But what I'm going to do now is show you a bit of a simplified filtering method that we're going to walk through for our case, really more as an illustrative scenario. And this filtering scenario is taken out of Nussbaum's textbook, of clinical genetics, and it's a representative filtering scenario that we might use or might use as a basis in our patient under study, where we find 4 million variants, and if we exclude those variants that are not within or near an exon, that might leave us with, for instance, about 80,000 variants. And we don't think that our patient has a common mutation. We suspect that if he had had a common mutation, we would have seen it. You know, we would have seen it. It would have been on the CHIP test. So people would have known about it. So we're assuming that our patient has a private mutation. So we can remove variants that show up frequently in public databases, and that leaves us with about 1,500 variants. From there, we can filter that a little bit further. We may not want to look at mutations that result in a synonymous change that may not result in any change in the amino acid, or that may not affect messenger RNA splicing. That leaves us with about 200 variants, of which possibly some of those will fit the suspected inheritance pattern. So I'm not going to dwell any more on this picture other than to say I'm going to kind of sort of follow this model. It's a little bit simpler than the New England Journal of Medicine model. I'm going to kind of sort of follow this model as we now begin to walk through the findings from our patient, and we're now going to try to whittle down his list of 4 million variants to see if we can ultimately figure out what the heck is causing his problem. So let's assume that our patient has 4 million variants, okay? And that's a lot. So we're going to filter that to those variants within or near an exon, and we wind up with about 80,000 variants. And you may remember back in deck number 4 when we talked about polymorphisms, we saw that a typical person will have about 3 million SNPs, of which about 100,000 of them are in exons, okay? So what we're looking at here is not just SNP variants, these are all variants. Some of these are SNPs, some of these might be copy number variants, some of these might be insertions, deletions. But when we filter all of the SNPs to just those that are within or near an exon, we'll find ballpark about 80,000 SNPs. Excuse me, about 80,000 variants. Now, again, we're suspecting that this person has a private mutation, and we're suspecting that he has a private mutation in the LDLR gene. And so we can go to public databases. There's one called the 1,000 Genomes Project, and then there's another one called the EXACT Project, where they've done either SNP-based testing or whole genome sequencing on lots of people, thousands of people, and they publish all of the allele frequencies and the SNP frequencies and the variant frequencies. And if we go into these databases and we say, okay, I want to filter my list of 80,000 variants, I want to remove all those variants that are common. I want to be left with the list of variants that show up less than 1% in all of these public databases. Then I'm going to wind up with now a list of only 1,500 variants, which is still a lot, but it's a lot easier to manage than 4 million. Okay, so now we got about 1,500 variants. But that's still a lot. We can further filter that list to variants predicted to be significant. And in this case, what we're doing is we're tossing out all the variants other than missense mutations, mutations that change the amino acid sequence. We're going to keep nonsense mutations, ones that introduce a stop codon. We're going to keep frameshift mutations, and we're going to keep mutations of a known splice site. Okay, so missense, nonsense, frameshift, and splice site mutations we're going to keep. All the other variants we see, synonymous SNPs, those we're going to exclude, and realizing, of course, every time we filter that we may have just filtered away the causative mutation, and realizing, of course, that we may only have a roughly 25% chance of finding the causative mutation in this approach we're going through. But still, this is a nice kind of way to start, and if it yields the answer, well, then that's great. So we're now left with about 200 variants predicted to be significant. There's a couple of tools that are available. This one is Ensemble's Variant Effect Predictor. You can feed it with a list of variants, and it will predict for you whether or not those variants are likely to be significant or not. And they have a number of different predictions that they do, so you can decide which predictions you're interested in. In our case, we could use something like this variant. We could feed our list of 1,500 variants into Ensemble's variant effect predictor, and we could simply say, return back just the ones that are either a missense, nonsense, frameshift, or mutation of a known splice site. Other people might want to say, well, I want to apply slightly different filtering, but the Ensemble variant effect predictor is a nice tool for allowing you to feed it a whole list of variants. In fact, you can just feed it a VCF file for a list of those 1,500 variants, and it will catalog each one as to the possible clinical significance. Now, you have to take these predictors with a big grain of salt, and because oftentimes these predictions are wrong, and these variant effect predictors are not anything that you would want to apply directly in a clinical sense. You wouldn't want to say, oh, well, the variant effect predictor said that this was a non-pathologic variant, so therefore it's non-pathologic, or the variant effect predictor said that this is a pathologic variant, so therefore it's pathologic. You certainly can't use them this way. The way we're using them is as a filter to then further explore the subset that we're interested in, realizing that we may have just removed the actual culprit. There are a couple of other, this is an example of how wrong these predictors can be. SIFT and polyphen are predictors that look at changes to amino acids. And they look at homology, and they look at phylogenetic trees, and they say, well, this is an amino acid in a highly conserved spot, or this is an amino acid that we see has been changed quite a bit through evolution. And so that it has a number of logic steps to kind of predict the clinical significance of a particular change in a protein sequence. And in this particular case, we're looking at sickle cell anemia. And in this particular case, neither SIFT nor polyphen predicted it to be deleterious. They said, so this is a non-synonymous change. This is a missense mutation where there is a change in the amino acid, but these two software programs did not predict that this particular change would be deleterious. And we were a little bit more conservative in our filtering approach. We simply said, look, if it's a missense or a nonsense, we're going to keep it in our subset. And so we're not really relying in our subset on the output of SIFT and polyphen. Just because, as shown here, it's very tough to rely on them from a clinical perspective. So based on our filtering for which variants we believe to be clinically significant, we then further filter one last time, and we say, okay, of those 200, which one of those variants is in one of the 4 known familial hypercholesterolemia genes, and which one segregates in an autosomal dominant pattern? So what we actually did here was we also tested the brother and we also tested the mother. Now, we could have tested the unaffecteds as well to gain additional statistical power, right? But here what we did was we looked in these 4 genes and we said, find a mutation in one of these 4 genes that this patient has, and we want to also see that same mutation in mom and brother. And when we applied that last filtering step, we were left with just 1 mutation. We started with 4 million, we worked down this filtering, and we wound up with 1 mutation. Okay? So now the question is, did we find the right one? How do we know now that this presumptive variant is pathogenic? So here's the mutation that we found in this particular patient. We found that in the LDLR gene at position 19, there's normally an A, but in our patient, it was changed to a T. So let me see if I can orient you to this picture. This is the first exon. This is the first exon of the LDLR gene. It's got a lot of different transcript forms. That's why you see it repeated so many times. But this is the first exon, and here's the first intron. So we're zoomed in quite a bit to this particular gene. Here is the start codon, right? The ATG start codon and the corresponding methionine amino acid. This first row shows the codons for the bases. At position 19, we see an A, okay? And in our particular patient, that A was mutated to a T. And that means this particular codon changed from an AAA, which is a lysine, to a TAA. And a TAA is a stop codon. So very early on, there's a mutation that changes this particular codon from a lysine to a stop codon. Okay? So we have a presumptive variant we've seen in this patient, and now what we want to try to figure out is, well, has anyone ever seen this variant before, and is this a pathogenic variant? And by the way, this is just a made-up variant. As far as I know, this is not a known variant. It's really just for the sake of our case and our example to walk you through the process. So we have a presumptive variant, and we know that it's a nonsense mutation. And the question is, has this variant been seen before? Okay. And I want to introduce you to a couple of new resources, at least one new resource. And the resource we haven't looked at before is ClinGen. This is another resource from NCBI, well, at least it receives its funding from NCBI. But the difference of this resource, in my opinion, from the resources we've looked at before, is that this is one of a couple of resources really being designed to support the needs of frontline clinicians. Okay, and if you look at their little graphic, really what ClinGen is trying to address is this a gene that's associated with a disease? Is this variant causative? And is there information that's actionable about this? What they're trying to do is design a utility, a genetics utility, that would be useful to frontline clinicians. And it's a relatively new resource, in my opinion, this is one to watch. With the pharmacogenomics discussion we had last week, there was the Pharm GKB and the CPIC websites. Those were being intended for clinicians, and this one's being intended for clinicians. And a lot of the other resources that we've looked at are for informaticists and bioinformaticists. So if we look at this website, we have a question about, has the variant been seen before? And so we can come down here and we can look at the allele registry. Okay, and so they have an allele registry here. And if we go to the allele registry, and basically what I'm going to do now is I'm going to just walk through the steps on this slide, okay? So let's look for the variant that we found. Here's the variant that we found, and let's search the registry. Okay, so now we have, and ClinGen has its own internal allele identifier, okay, which we're going to need to do, which we're going to need to obtain in order to explore ClinGen resources. So we now have the allele identifier for the particular variant. And now what we want to do is look at their variant curation tool. Okay. And so let's now take a look at this particular variant. And what's nice about ClinGen is it's going out to the 1000 Genomes project, it's going out to the Exact project, it's going out to a lot of different places. Oops. It's going to a lot of different places that I don't have to go look up all that information myself. So this is the variant that we found in our patient. And one of the questions we have is, well, has it ever been seen before? And what we're finding here is that it's never been seen before in ClinVar. And when we look in these population studies, it's never been seen before in Exact. It's never been seen before in the 1000 Genomes project. This is a variant that we've found in our patient, basically, where as far as we know, it's never been found anywhere else. Now, keep in mind, you know, there's a lot of other websites out there that are keeping track of variants, and it's not as though this is really a comprehensive source, and I'm just kind of sort of doing this for sake of illustration. And I imagine that this website is just going to get better and better, and that using ClinGen, that a clinician over the course of the next couple of years can, with greater and greater reliability, use ClinGen as their single point of searching to see whether or not a particular variant has ever been seen before. Right now, that's not the case. You know, you still have to go poke around a little bit if you really want to. But for now, let's assume that the variant that we found in our patient has never been seen before. It's not in ClinVar, it's not in Exact, it's not in the 1000 Genomes. It's as far as we know, it's brand new. Okay. Okay, so there's no data on this variant found in ClinVar, Exact, or 1000 Genomes. So the question is it pathogenic? And what we're going to look at here, in order to assess the pathogenicity of a variant, we're going to be looking at the American College of Medical Genetics and Genomics guideline for interpreting sequence variants. And by the way, we're looking at this article. I put this article on the download site, same place we keep the PowerPoints. The URL for this article is in the PowerPoint also, but the article itself is there. And what we're looking at specifically now is kind of like a summary table out of this guideline. And what they try to do here in ACMG guidelines are come up with a way that we can consistently across labs and across research settings determine whether or not a variant is pathogenic and speak the same language, so that if two labs report a variant as pathogenic and they both say it's based on ACMG guidelines, then we have a level of confidence in that. And in addition to that, we can point to the specific evidence for pathogenicity. And the way that the guidelines work is they examine a lot of different types of evidence in order to determine pathogenicity. And then there's a scoring mechanism, and there's a calculator that can be used. And so population data, for instance, we see that our particular variant is absent in population databases, okay? And that's moderate evidence of pathogenicity. We anticipate that this is an autosomal dominant disease. We would think that if we found that this variant was prevalent out there in patients that didn't have any phenotypic signs of familial hypercholesterolemia, it'd be kind of surprising that that was the culprit variant. But in this case, we don't see that variant out there, and that provides moderate evidence. We have very strong evidence of pathogenicity based on our computational and predictive data. And despite what I said about the potential shortcomings of things like SIFT and polyphen, in this particular case, we know that we have a null variant. We know that the particular mutation in this patient has changed a lysine to a stop codon. And we know that there's other variants in the LDL receptor gene that result in disease. So we know that we're dealing with a gene where loss of function is a known mechanism of disease, and we know that this particular patient has a predicted null variant. So based on the particular mutation we're seeing, we predict that this is pathogenic, and that's very strong evidence for pathogenicity. Functional data would refer to, for instance, lab data or something that's done in vitro. It's some testing of this variant, and we don't have any functional data for this particular patient. We do have segregation data. Segregation data means that in our case, we tested the patient, his brother, and his mother, and we found that all three of them had the disease and also had the particular variant. Now, if we had tested even more family members, if we had also tested that the father didn't have the gene, or that the unaffected sibling also didn't have the gene, and if we had tested, the uncles, etc., then we would have greater and greater segregation data. And the guideline's a little bit fuzzy here, but the more segregation data you have, the stronger the statistical power is of your segregation data, the more you can use that to assert pathogenicity, okay? De novo data doesn't apply here. We're not looking for a de novo mutation. Allelic data doesn't apply here because we're not looking for a recessive disorder. We're assuming that this is autosomal dominant. We don't have any evidence, we don't have any information about this particular variant in other databases, and we don't have any other data. So the main pieces of evidence we have in our case in determining pathogenicity are the ones circled here, and we can now plug this in. We can now plug this in to our clinical pathogenicity calculator. Okay, and let's just do that because it's kind of groovy to kind of see what happens. So let's go ahead and here's the variant that we found. and this is the allele identifier. Okay. And, oops. Okay, so let's find that. Well, it's not wanting to find it. Let's search for this instead. Okay, so we're going to search by HDVS. Oh, want me to log in again? Okay, so here's our variant. Okay, it was registered in ClinGen. It has its allele identifier. Now we're going to calculate. Okay. And the calculator is kind of cool. It allows you to plug in the evidence. And the reason I think that this is important is because, okay, you look at this and you say, okay, so we have some supporting evidence, we have some moderate evidence, we have some very strong evidence. But what does that mean? Do we have enough to assert that it's pathogenic? But when we go to the calculator and we now say, okay, we have moderate evidence, here, we have population data, we have computational, we have very strong computational data. And we have supporting segregation data. Okay. And let's see what happens. Okay. The calculator has deemed that this is a pathogenic mutation. The criteria is as long as it has at least one moderate, one supporting, and one very strong. So now, based on the American College of Medical Genetics guidelines, we've concluded that the variant that we've identified in this patient is indeed pathogenic. As a final step, what we would do, which I'm not going to do here because this is fake, but if this were real, what we would do is we would submit this variant to ClinVar, so that then this information on this particular variant is then available for others. And when you wind up submitting to ClinVar from here, all of this supporting evidence winds up getting stored as well. And so the ClinVar submission would say, okay, here's who submitted this information, and this was the supporting data, so that then others could look at that particular. You may remember from ClinVar, oftentimes we saw conflicting evidence, and this is a way of trying to help resolve the conflicting evidence in ClinVar. Okay. And so now we register the variant with ClinVar. Okay. So we've now identified the mutation that the patient has, and we've assessed it for clinical significance. We believe that that's the one. And what happens then is, finally, when labs are reporting the results of next-generation sequencing, they are reporting secondary findings as well. And here again, the American College of Medical Genetics and Genomics has come up with a list of secondary findings that should be reported. And so, for instance, if there is a pathogenic mutation of the adenomatous polyposis coli gene identified as an incidental finding, that should be reported, even when you're reporting, when you're reporting other stuff as well. And so they maintain this list of secondary findings that will get reported as well. And then that kind of completes the end-to-end process. And that is it, other than I want to have a little bit of time left to go over the homework assignment. So let me just pause there for a minute and see, does that kind of sort of make sense? Any questions? It's kind of interesting, kind of complicated, the way that the whole process works. I found that particular New England Journal article to be very interesting because they walk through this whole process and they actually use it, and they wind up diagnosing 25% of the patients that were referred to them. Some of you, because it's been in the news a little bit, may have heard about the Annals of Internal Medicine article that came out recent in the last edition by Vassi. The difference there was that he was doing DNA sequencing on patients without any symptoms. He was doing it as a screening maneuver. Here, we're doing the DNA sequencing on someone that has a known phenotype. And so that makes a big difference in trying to hone in. In fact, when you look at the American College of Medical Genomics guidelines, they're very clear that their guidelines for determining pathogenicity can only be applied in the setting of a phenotype. And so their guidelines would not be applicable, for instance, to Vassi's study, where he was doing DNA sequencing as a screening against a healthy population. Okay, you guys ready for your homework assignment? Okay, this is going to be the hardest one. And I would really encourage you guys to collaborate on this assignment with one or more classmates, because I think at least some of these are going to be hard. And I would also encourage you, if you're really trying to work the homework assignment and you bump into just a roadblock and you're getting frustrated, to please call me or email me so I can help you through it. Here's the scenario for the homework assignment. You've identified the following germline variants, okay? And there's 4 of them. The fourth one, in fact, is just the example, the fire example. And what I want you to do for your homework assignment is for each one of these 4, figure out what's the gene in question, is the variant pathogenic, what disease, if any, is the variant associated with, and where did you find your information? Okay? And I will tell you that some of these are going to be easy and some of these are going to be hard. Okay? And so I think because of that, it's going to help to collaborate with one or more of your classmates, because some of you may remember one resource, some of you may have remembered another resource. Plan to spend about 60 minutes. In 2 weeks, when we cover the clinical genetics informatics session, we're going to go over this. And here's some helpful hints as to how to even begin to approach this particular assignment. And by the way, I think that the hours spent on this, the poking around, and the familiarity that that's going to give you with the informatics and the bioinformatics resources, it's designed to be a little bit of a difficult homework assignment, not straightforward, because it's going to force you to have to get to be a little bit familiar with some resources that I believe are going to be quite valuable to you. So here's a helpful way to begin your searching. To start, convert variants into HGVS syntax. That's the way you want to search. The first 3 are already done for you. The 4th one would have to be converted into HGVS syntax. Then, using your HGVS syntax, begin by searching large, well-known repositories, such as ClinVar or dbSNP. Okay? Keeping in mind that you're not going to find any comprehensive repository out there, If you don't find these variants there, then you can start checking locus-specific databases as needed. And again, there's no comprehensive locus-specific database, and so it's possible that there's going to be a variant and you're simply not going to find it out there, even though someone may have already discovered it. But there's a large directory of locus-specific databases here. And then from there, if you still can't find it, you can try to predict the pathogenicity either using a tool like VEP or ClinGen. Okay, and if you run into problems, if you get frustrated, again, just reach out to me, let me know, email or phone, and try to collaborate on this. And I look forward to seeing what you guys come up with. Do you guys have any questions about the homework? Okay. Are you guys going to do the homework?
Yes.
That's right.
Okay. All right.
I think everybody is on mute.
I know everyone's on mute, but I captured the names of everybody on the call.
It's recording, so.
I'm going to let Payal do it with me.
Good. No, that's great. That's great. Yeah,
Sure.
I would really encourage you guys to work together on this. And I do think that some of the examples you'll find are easy and some of them are hard. And I'm certainly happy to help if you're struggling with it. Just feel free to reach out to me. And then I think that's it for today. The only thing that I would like, if you guys wouldn't mind, is to just tell me a little bit about today's class. Too much, too little, too fast, too slow, just right, too hard, too easy. Any feedback today?
Yeah, it is a lot of
Okay.
information, but I also came late, so I missed some of it. But I like the weaving in of the informatics stuff.
Oops, oh wait, this is backwards, isn't it? Okay. Anything else? All right, guys. Thanks a lot. And next week is genetic engineering. Have a great week.
You too.
You too.
Take care.
Thank
Bye.
you.